Hello children, let us learn more about the fundamental unit of life, that is cell. And in this lecture, we are going to start with the topic transport of various substances across the cell membrane. So, how various substances required for the survival of a cell cross the plasma membrane. There are various modes such as diffusion, osmosis, endocytosis, and active transport. Let us understand them one by one. So in diffusion, by definition we know it is the spontaneous movement of any substance from the region of its high concentration towards the region of its low concentration with or without any semi-permeable membrane in between. But here in case of cell, there will always be this plasma membrane present in between. So what are those substances which enter or exit the cell through diffusion? Gases, essential gases like CO2 and oxygen. Now we have to understand where the relative concentration of CO2 will be more and where will be the relative concentration of oxygen in higher concentration. So remember, inside the cell the oxygen is continuously getting depleted and CO2 is continuously getting generated. So CO2 will diffuse out of the cell while oxygen will diffuse inside the cell. This is how these essential gases will enter or exit the cell. Now let us talk about osmosis. So osmosis by definition is the spontaneous movement of water molecules from its high concentration towards its low concentration. And as we have understood this in detail also, that concentrated solutions will have less water concentration. So osmosis can be of two major types depending upon whether the water is entering inside the cell or leaving it. So what is end osmosis? If the cell is placed inside a hypotonic solution, then the total movement of water molecule, that is net movement, will be towards inside the cell. Remember, water will leave the cell as well as enter the cell, but more water will enter the cell than will leave it. So as a result, the total volume of cell will increase or the cell will swell up. Now what is exosmosis? Exosmosis is observed when we keep the cell in a hypertonic solution. So that outside solution is having less concentration of water. That's why the cell starts losing the water. By which I mean more water leaves the cell than enters the cell. So net movement of water is towards outside. As a result, cell shrinks. So here you can see they are showing a plasmolysed plant cell. So the shape of cell wall did not change, but you can see that the entire plasma membrane has shrunk because the cell has lost a lot of water. And then there is another situation. What if we are keeping a cell in a solution which has exactly the same concentration as that of cytoplasm? So equal amount of water will enter the cell as will leave the cell. So net movement of water becomes zero and the volume of cell cytoplasm doesn't change. So in this condition, cell remains flaccid, neither plasmolysed nor turgid in case of plant cell, neither crenated. This shrunk stage of animal cells is known as crenated stage, and if the endosmosis continues for a really long time, the cells will burst. So here you can see they are showing some loss of the cytoplasmic material. So this is how we understand that in which direction the net movement of water will take place. Now we have understood how the gaseous and water molecules enter or exit the cell. What is endocytosis then? Well, it is a way to import or expel solid materials. So in this, remember children, the flexibility of plasma membrane plays a very important role, because that is how a small vesicle is created around that particle which is to be imported. So endocytosis is something that we can never observe in a plant cell, because the cell wall is a rigid structure. We need the flexible nature of plasma membrane to take over, only then this is possible. Now, so far you might have noticed that in diffusion and osmosis I had used the word spontaneous movement, by which I mean that there is no expenditure of energy involved. But there are certain substances which need to be actively transported in or out of the cell, and there are certain conditions where we need to defy the concentration gradient. So, for example, we need to send some molecules from their low concentration area towards the higher concentration area. Since this is against the concentration gradient, we need to spend energy. So whenever it is active transport, remember, expenditure of energy in the form of ATP will be there, and that is why whenever we spend in energy, then we call it active process. With this, we have understood that how the cell membrane ensures that the cell has got everything it needs. It will utilize all these processes to import or expel the substances according to the requirement of the cell. Now, let us learn about an additional covering, cell wall. Now, this is not a universal covering as in case of plasma membrane. Remember this point, because plasma membrane will be present in all those structures which are to be called as cell, but cell wall is found only in plants, bacteria and fungi. So this is not a universal covering. Where is the location then? It lies outside the plasma membrane. So a cell, like a typical cell, will have cytoplasm covered by the plasma membrane, but cell wall lies outside the plasma membrane in plants, bacteria and fungi. Now what is the nature then? It is a rigid layer, so it helps the cell to maintain its shape, and it is freely permeable. So both the things are very different from plasma membrane. Plasma membrane was flexible and it was semi-permeable, but cell wall is rigid and freely permeable. But then why is this important? Why is it present? Because in plants, bacteria and fungi, they need to withstand hostile environments. Suppose sometimes the environment is very dilute. This situation will be like hypotonic solution, and cell will experience endosmosis. But the cell, like for example plants or fungi, they cannot move just like animals. They have to be there and sustain those surroundings. So what happens, children, after a lot of water has entered the cell and it is swollen, it is turgid, that extra pressure that the cytoplasm is applying on the plasma membrane is now neutralized by the equal and opposite pressure of the cell wall, and cell does not burst. So the main important feature of cell wall is that it helps these particular organisms to withstand dilute external surroundings. So that's how they tolerate greater changes in their surroundings, because they are immobile and they need to come up with some kind of solution where they can survive in the exact place where they are. But animal cells would have burst, or they might have moved away from the dilute surroundings, as the case may be. So cell wall is found only in plant, bacteria and fungi. Remember that. Moving ahead, now we have come to a very important part of any cell. So let me make it very clear. By nucleus I mean the part where nucleic material will be present. Now this part is found in both the cells, whether it is prokaryotic or eukaryotic. Nucleic material will definitely be present in both types of cells. What will be the difference? That in prokaryotic cells, the nucleic material lies just in a particular region of the cytoplasm known as nucleoid, while in prokaryotic cells, this nucleic material is enclosed in a double membranous protective structure. So it is a very distinct cell organelle known as nucleus in the eukaryotes, nucleoid in the prokaryote, where there is no membrane covering the nucleic material. So let us talk about a typical eukaryotic nucleus. A nucleus is a double membrane bound structure present in the cell, and it is a very prominent structure. It has two layers of plasma membrane, and this layer has got pores in it. Okay, what lies inside? So just like the cell has cytoplasm, even inside the nucleus there is a nucleoplasm. What else is there? There is an entangled mass of threads. So this entangled mass is actually long lengths of DNA which constitute the genetic material, and we call it as chromatin. And what else do we see? There is one particular area within the nucleus which always stains darker than the rest of the portions. This portion is nucleolus. So what nucleolus is involved in, that from all the regions of this chromatin material, different RNA will be formed as the cell requires, but all that RNA which is going to form a ribosome is known as ribosomal RNA. Ribosomal RNA will be gathered and assembled here along with certain proteins, and once nucleolus has collected ribosomal RNA and essential proteins, these parts will be exported out of the nuclear pore into the cytoplasm, where the ribosomes will be completely assembled and ready to function. Remember, ribosomes synthesize protein, but their own synthesis is taken care by nucleolus, halfway here and remaining half in the cytoplasm. Now it is time for us that we clear all the doubts about chromatin, DNA, chromosome, gene, etc. So here is one more diagram which will make your doubts clear. Suppose this is any cell and this white portion is representing the nucleus. So I said that there is an entangled mass of DNA which is known as chromatin. So what is this rod-like structure joined to each other in the shape of X? Well, this also is made up of chromatin, but that chromatin which has been condensed in a very systematic way, giving rise to these rod-like structures. So why didn't we observe this kind of structure in this nucleus? Because children, when the cell is in its normal metabolic phases, non-dividing phases, the chromatin lies like this. It is only when the cell decides that now it is time to divide, before division the chromatin will be condensed and they will form distinct chromosomes. Now how many chromosomes will be present inside the nucleus? That depends on different kinds of organisms. Let me give you one example, that in human beings, inside one nucleus you will observe 46 such rod-like chromosomes, and in a tomato cell you will observe 24 this kind of chromosomes, and so on. Different organisms have different number of chromosomes, and I would like you to find out which organism has got highest chromosome numbers. Now, moving ahead, DNA is making the chromatin. Chromatin condenses and forms chromosomes. Now, is there something more to be learned? Yes, there is. Ultimately, you might have understood that this chromosome is made up of chromatin, which is made up of DNA. Now, this DNA is a double stranded structure, by which I mean two distinct strands are coiled on each other. And suppose this particular portion of DNA from a particular region till a particular region decides a particular character. For example, your skin color, your hair color, and the shape of your nose, various metabolic activities of your body. Another portion of the DNA will govern different functions of the body. But remember, that particular portion which governs one feature of the organism is known as the gene. So now we have learnt many important things about nucleus, which is also known as the brain of the cell or command center of the cell. Let us summarize one more time. So nucleus is a double membrane bound structure. Yes, it is. And its nuclear membrane has got pores. So pores are present there. For example, you have seen that ribosomal RNA and proteins will exit the nucleoplasm and go to the cytoplasm. Also, various RNA which are formed inside the nucleus have to be sent to the cytoplasm, where they can form different kinds of proteins with the help of ribosomes. Now, nucleus also contains genetic material or nucleic material, which is made up of DNA. Nucleus of a non-dividing cell will show the DNA as an entangled mass known as chromatin material. Now, chromatin is made up of DNA and few proteins, very specialized proteins, which help to package the DNA within such a tiny space. You know that eukaryotic cells range from 5 to 100 micrometer. But just for your information, a human cell, a single human cell might be having the DNA up to 2 meters long. And now you can multiply this 2 meter with the approximate number of cells in a human body. So you can imagine that 2 meter long DNA can be accommodated in such a tiny cell which is not visible with the naked eye. Then how? All that packaging is possible due to certain specialized proteins. So chromatin is made up of DNA and those proteins. Now when the cell is about to divide, chromatin materials gets systematically organized and condensed into rod-shaped structures known as chromosomes. And the number of chromosomes will vary in one type of organism from a different type of organism. Now chromosomes, once more, remember, are composed of DNA only and protein, and they contain information for inheritance of characters from parents to next generation. Now where is this information present? It is coded on DNA in the form of gene, as we discussed. That particular region of DNA which codes for a particular kind of feature is known as gene. So ultimately we have understood why this nucleic material is so important, because the parental characters will be transferred to the next generation through chromosomes only. And functional segments of DNA containing the information necessary for constructing and organizing the cell are called genes. So there will be thousands of genes governing each and every aspect of a cell, its appearance, its metabolic activities, everything. Thus, let us summarize what is the function of nucleus. It plays a central role in cellular reproduction or cell division, and it also determines all aspects of cell development and its metabolism. So this is about nucleus. Now we understand why is this known as the brain of the cell. Moving ahead. So let us talk about cytoplasm. This is no ordinary liquid. It is a wonderful liquid because it is the gelatinous liquid which is filled inside the cell. If this is a liquid, why is this so special? Because it contains all the essential salts and various organic molecules which make the cellular environment suitable for the cellular metabolic activities. Remember children, cell is the functional unit of life. So many living processes are going on inside the cell for which a particular pH, a particular concentration of various salts and nutrients, gases is required. And all that is present in the watery background of cytoplasm. And water constitutes almost 70% of your cytoplasm. What else is present in it? All the cell organelles are floating in the cytoplasm. So you can imagine that your cell is just like a water filled balloon, and in place of water there is cytoplasm, and in place of all the glittery materials, cell organelles like mitochondria, lysosomes, endoplasmic reticulum, nucleus, etc. are floating in that balloon. So cytoplasm is very important fluid of the cell. With this, remember, we have covered all those structures which are essential for any structure to be called a cell. What are those structures? Plasma membrane, nucleic material, which might or might not be covered by the nuclear membrane, and third, cytoplasm. In our next lecture, we will start learning about the cell organelles. But remember, revise all these topics very well and come prepared in the next class, where we will start with the endoplasmic reticulum. Thank you.